realize that I think you are not too familiar with chapter 21. So that's why I need to discuss with you, okay? The chapter 21. Okay, so you can see my screen right now. I'm showing your paper. Okay, so that's why explain, okay, they ask you to describe and explain the difference before and after in the salt solutions. Okay, so you wrote the keyword, which is this one, okay, turgid, flaccid, and plasmolized. But when you answer this kind of question, right, osmosis, okay, related to osmosis, and then you already know that the water molecule able to diffuse from the region of high water potential to the region of low water potential. So you should mention that which region have a low water potential, which region have a high water potential. So you answer, you already mentioned that, yeah, the salt solution, they have a low water potential, okay? But it will be better if you say that the salt solution, they has a lower water potential if they compare to the salt cytoplasm. Yeah, so by this way, right, you can I mean when the examiner they mark your answer, they can visualize that. Yeah, okay, so this is a cell. So you know that this cell, right, they have a higher water potential, right? And then outside the cell, yeah, which is the salt solution, okay? So here is the salt solutions. Salt solutions, they have a lower water potential. Yeah, they have lower water potential. So that's why the water molecule, they're able to diffuse from cell, I mean, from this cytoplasm of the cell to the outside, right? From the cytoplasm of cell to outside salt solutions by the osmosis. Yeah. So your answer should write in this way. The salt solution, they has a lower water potential as compared to the cell cytoplasm. Or you can say that the cell cytoplasm, they have a higher, they have a higher water potential than outside salt solution. So water, they able to diffuse, okay, from the cell to outside the salt solutions through the partial permeability. Okay, this is correct. Okay, I can see the see the keyword, which is a partially permeability. So that's why the cell lose water and then it shrink. Yeah, so this question, right, I mean this answer you can get 4 marks, but actually it's 6 marks. Okay, 6 marks. So if you write this one, you can get 1 more mark. Okay, like this I can get 1 more mark, and then yeah, okay, which is around like that, around like that. And then you should mention osmosis. Do you mention osmosis? Yeah, I didn't mention osmosis. So you should mention osmosis at here, okay? By osmosis. And then what happens if the cell, they lose water, right? The vacuum will shrink, and then what will happen? Not only the vacuum will shrink, and then slowly, okay, slowly, the cell membrane, right? They pull away from cell. So this is in the cell. Yeah. Okay, this is correct. Okay. And that's whenever you write about this one, muscle constrict. Okay, muscle, you cannot use this keyword constrict. It's not suitable. Constrict, right, you can use in where? Your pupil. So remember, your pupil can dilate and constrict. Let me show you. Where is it? Okay, here. I think it's here. So that's why you should use the important keyword to answer it. I think here. Yeah, let me show the chapter 14. Right? Chapter 14 will about the pupil. They will dilate and they will constrict. Yeah, so that's why the pupil, okay, when we go back to see the chapter 14. Okay, which is here. Okay, where is it? I think I got put the picture, right? The pupil constrict on. I didn't put it. Let me check. Okay. Oh, I haven't included it inside. Sorry. Yeah, I think at here, right? Chapter 14. Okay, let me show the Mandarin. Okay, I think I put in the Mandarin already. Which is this one. Okay, the pupil constrictions and dilation. Okay, so constrict we talk about what? Pupil size. Okay, so you can use this word constrict. You cannot use pupil contract. Wrong answer. Okay, you can see my screen, right? I'm showing the pupil side. You can see. Okay, I think I'm sharing. Yeah, I think I'm sharing. Yeah, so that's why pupil constrict or pupil dilate. And then you should know that why the pupil constrict is because the circular muscle. Okay, you have you have a pair of muscle, right? One we call as what? Circular muscle. Another one we call as the radial muscle. So when the circular muscle, they contract, okay, not constrict, contract, because you can only describe either the muscle contract or the muscle relax. So that's why you should mention that hair muscle, okay, and it's not hair muscle, okay, it's hair erector muscle. Your hair don't have muscle, okay, it's hair erector muscle. I need to see this erector, okay, so that's why this I cannot give you mark because you mentioned hair erector muscle, they will contract. So when they contract, okay, they will cause the hair to stand up. Okay, so when the hair they can stand up, right, they can trap the insulating layer of air. Okay, so this is too much. Okay, so I think later I will update the actual answer for every paper. Okay, and then you can spend some time to read. And then you are writing some of the point. Okay, it's very less the point that you write. You can write about what? You write about vasoconstriction, right? Okay, and then you can write about shivering produce a heat. And then what you can write. So this is happen that when you feel very cold, okay, when you feel very cold, so your body will take part to reduce the heat loss. Okay, so that's why whenever you feel very cold, okay, you can say body stop produce sweat. Yeah, okay, you can include this answer also because it will not produce sweat in the cold period. It will only produce a sweat in the hot day. Yeah, in the hot day. Okay, so this head the muscle, and then this one. Okay, the importance of water. This one support cell and tissue to function. It's a bit weird when I look at this statement. Okay, water, right? They can help to allow the chemical reactions to take place. Okay, it is the medium, medium for the chemical reactions. It can help digestion, excretions, and other kind of the body process. And then they can help to, most important, right? Especially in the plant cell. Yeah, because in the plant cell, you don't have the backbone to support the plant. So in the plant cell, they require the water. So water, they can help to maintain the shape. Okay, they can help to maintain their shape in the plant cell. Yeah, because the plant cell, they consist of vacuole. And then inside the vacuole, they will store a lot of water and mineral ions. So the vacuole, when they expand, right, they can press against the cell wall, creating turgor pressure. Okay, the turgor pressure. This is also correct. Okay, I think you shouldn't have any problem. Yeah, I think this one. Okay. This is, they talk about what? Plasmid energetic modification. Do you know where is the plasmid? Where you can find the plasmid? In which organism you can find the plasmid? Yes.
Bacteria.
Yeah, it's bacteria, right? So that's why usually, right, bacteria is a very, very suitable tools, okay, to involve in the genetic modifications because it will not have any, it will not create any ethical issue if you use the bacteria. If you modify the gene in bacteria to serve your purpose. But if you modify the genes of animals, right, or you modify gene of humans, right, and then a lot of the, people, they will shout at you because this is unethical. This is unethical to them, okay? But it's not too good also if you modify on the human gene. Okay, because you are unsure about the consequence. Okay, any further consequence happens if you modify the human genes, right? So that's why usually we are not allowed to do that. So you can modify on the bacterial genes, and then it's more easy to modify also because the bacteria, right, they have special structures only found in the bacteria, which is this one is known as plasmid DNA. Bacteria, they have two types of DNA, right? So remember the structure in the bacteria, they have two types of DNA. Okay, they just look like this. This is a bacteria, right? This bacteria, and then here will be the DNA. Okay, so this DNA is circular. Okay, this is circular DNA. Circular DNA. And then you have another ring here. So this one is called plasmid DNA. So that's why during genetic modifications, human and DNA and plasmid DNA are cut with the restriction enzyme. Okay, so restriction enzyme is a scissor. They help to cut part of the gene. So that's why they will just take out this bacterial plasmid, right? And then they will use this enzyme. So this enzyme to cut on the certain region on the plasmid DNA, and then they will just create a sticky end. This one. So this is not a sticky end. What's meant by sticky end? It's just like after the portion of bacterial plasmid is being cut, being removed, right? So they will just like this. So this region, right, which is don't have any stuff inside, we call as what? We call sticky end. Okay, sticky end. So you have a sticky end produced in the plasmid DNA. You also have a sticky end produced from the host DNA. So that's why they can join together by the ligase. So we'll see this word ligase, right? So ligase GG is grouped together. So it's just like they will tend to combine two different molecules together, become one complete molecule. So G together, okay, means group all together. So the modified plasmid contain the human genes called as the this one recombinant plasmid. Okay, and then this one discussed with before, and then just refer to the answer. Okay, just refer to answer, but you're lacking on some point. So that's why you should write the answer, which is totally fixed to the point. And then this one, okay, analytic set and then fluid, right? The functions. So their function is to what? Allow fetus to develop muscle and bones. This is not correct, okay? The second one is not correct. It's because actually how they help the fetus is in the indirect way. Because muscle and bones, right, they will always grow. When they grow, they need the space. So that's why this analytic set and the fluid, right, will provide the baby with the space. So that's why they will have more space to grow, develop their muscle and bone. And then this one also correct. Okay. It's not too hard, right? I hope it's not too hard for you. Explain one other adaptations of the group of aquatic plants. Okay. So you write about this one is correct. Stomata on upper epidermis of leaf, okay, on leaf, but you should write your descriptions, okay? So like in the second question, right? Okay, in the second answer, you do write about the point and then the description. So that's why you don't need to prevent the water loss. So that's why the stomata is on the upper epidermis of leaf in the aquatic plant. Because this is to prevent, okay? You can add the descriptions to prevent the stomata immerse in the water. So what happens if the stomata immerse in the water, right? The gaseous exchange will be inhibited. Gaseous inhibited. And then this is also correct. Okay, I think other than that is correct. Yeah, that's all. So we will continue discuss about chapter 21. Okay, so this is chapter 21. Okay, and then I want to show some of the questions for the biotechnology, right, and the genetic engineering. Okay, so I also have sent you the notes. Okay, these are the notes. Okay.
Wait, sorry.
Yes.
Could you remind like the boundaries for A star for paper 2, 6, 4, and 6? out of 80 and 40.
It means the mark boundary.
Yeah, so paper 2 is 36 out of 40. What is it for paper 4?
You mean for A or A star?
A star.
A star, 70 out of 80. It better you can get, okay? 70 out of 80.
Okay.
I think I said in group before. Okay, let me check. For the mark boundary for the A star, right?
Yeah.
Okay, let me show you again. I think last time I write down at here. Where is it? I think I write down at here. This one, okay. So you can refer to here. Okay. Yeah, you can refer to here, okay. But actually the mark boundary, right? The mark boundary is like they will be different for every year. But I think last year the mark boundary is quite high. Actually, it's the highest compared to the previous year. So that's why a lot of students, right, they will, they think that they can get A star, but actually they get A like that. So that's why it's better if you can hit for the higher mark, it will be better for you, okay? So for example, the paper 2, okay, the paper 2, so you have 40 question, right? Okay, and then you get how many? For example, you get 35, okay? For example, you get 35 times 1.5. Okay, so you get 48, okay? This one, 48 out of 200, because 200 is a total mark. And then paper 4, total mark is 80. Total mark is 80, right? 80 times 1.25. Okay, so this is the request what? Weightage. Okay, this is weightage. And then you get this one, 80 times 1.25. And then later you will need to divide by 200s. Yeah, you need to divide by 200s. Okay, so this is for the paper 4. 80 times 1.25. Yeah, okay, you get 100. So if you got, and for example, if you got 65 out of 80, right? Okay, so that's why 65 times 1.25. You get 81.25 out of how many? 81.25. Yeah, okay, out of the 100. Okay, so this is for the paper 4. This is for paper 4. Because later you need to add on. You need to add on this like 48. Add on the 81.25. Okay, like 48. Okay, you add on 81.25 and then plus your paper 6. For example, if you get, 35. Okay, 35. So means that 35 you need to times 1 only. Okay, so 35 over 200, right? So that's why you need to add on this one. Okay, you add on the 48, you add on this one, which is just to get 65, right? Okay, you add on 81.25, and then later you add on this 35. Okay, you get 164 over 200. So this is the total mark. Okay, so if you get 164 over 200, right? So your range is around 8. Okay, 8. So if you want to get the A star, so if rather you can get 70. Okay, 170. Do I have the last year mark boundary? Let me find. I think I didn't say for the last year mark boundary. Okay, so it's just like this. Okay, so at this point now you know how to calculate it.
Yeah, thank you.
Yeah, so that's why so that's why I say that if you want to get A star, right, so you try your best to score in paper 2. Actually, paper 2 is very easy to score. You just need to ensure yourself. Okay, so I think because we don't have much time, right, and then if you're able to do the cell division, so later I'll upload the paper 2 questions. Okay, I'll upload from 2023, 2024, and 2025. I will upload this 3-year questions. So you have total how many questions to do? 21 paper. But I think last time we have to few of the question already. So it's about 15 paper you need to do. You just need to do first, and then you mark by yourself. Okay, you can try to mark by yourself. The important thing is you just need to like look at the trends on the questions. Okay, look at the trend of questions because they will always come up in the similar question. Okay, similar question. So this is the A star boundary. Okay, which is 65. For example, okay, if paper 4 you get 65. Okay, you try your best to get 65. I know if 70 over 80 will be very hard, right? But we'll try our best to get 65. And then so this is the mark that you get. And then paper 2, right, you get about how many marks? 35. This one 35 over 40. And then paper 6, okay. If you're able to get about at least you should get 35 over 40, 35 above, 35 above. So yeah, so that's why if you get 35 above, right, okay, so 81 and then plus the 52.5, and then you get 35. Okay, yeah, 52.5. Yeah, you get total is 168, 168, which is a bit just 2 mark further away from the 1670 only. Okay, but it depends. Okay, sometimes it will be lower, like 165 over 200, right? You can get A star. But I think last year, right? Last year they put 170. Yeah, last year they put 170. So 160 above will be A. So 170 will be A star. Okay, so this one 170 will be A star, and 160 will be A. I think last year it's like this. And then if you get 150, right, you get B. Okay, so that's why paper 4 is very important. Okay, yeah, and then as well I have uploaded the paper. Okay, so this one, right, this one, paper 4 keyword analysis. You can spend some time to read it. And then most important is to read on how you're able to write for the paper 4. Okay, you can read on the answer. You read on the answer, and then you familiarize with the keyword. Okay, and then I have uploaded paper 6 for you. Okay, actually I uploaded the answer already. Okay, this is last year, which is include 7 paper, right, from the March, May, and the October. Okay, so you can have a read. I want you to read first and then see the trend. Okay, see the trend, how to answer, and then how to write a table, how to draw a table. Okay, write the answer. So that's why you need to read, and then you try to do. I think you try to do at least 2 questions. Okay, 2 questions. I mean, it's 2 paper from 2024. Yeah. So later I just send you. You read first. Okay, you read first. Read on how to answer it because it's just very simple. Paper 6 is the one with the easiest to score, easiest to get mark because you are not required any technical knowledge. You just need to memorize some of the keyword. Okay, and then you need to identify the variables or the variables, the kind of thing. And then most important that you need to able to know how to draw the image. Okay, so at this one, can you help me to draw the image? Okay, yeah, based on the I mean based on the this one. So this is already written in the answer, right? So they will ask you to draw a large clear outline using the pencil. Okay, so you should use the pencil to draw it, and then just draw the outline. That's all, just a very simple outline, and then just ensure that it's very larger than the original photograph. So so this is your drawing space. Okay, you should draw as large an image as you could. Draw, I mean, at least 3 out of 4 of the space is being used up. And then some calculation. Yeah, this is some calculation. So this is, I think you can have a read. Okay, have a read. For the paper 6, right? Do I mention it here? Paper 6. Yeah, paper 6, right? They like to come out in this 4 topic, which is the osmosis, the food test, the photosynthesis, and the enzyme. So these 4 topic. Okay, especially the food test, right? The food test, the osmosis, okay, they will come out the potato strip, how the potato strip change their length. So when they absorb the water, they will increase in their length or increase in mass, or they are losing the water, right? They will be decreasing in mass and also reducing in their length. The food test, okay, they will ask about the food test result. They will ask you to conduct the food test experiments. For example, use the Benedict solutions. You need to heat up under the Bunsen burner. And then photosynthesis, right? They will always ask you to calculate the rate of photosynthesis. Like what kind of factors? Okay, what kind of factors they will affect the photosynthesis? For example, the light intensity, the carbon dioxide concentrations, okay, and then the enzyme. Yeah. So generally, this 4 topic is very common. Okay, so yeah, later I will upload the paper too, and then you try your best. You mark it, and then you correct by yourself. Okay, you correct by yourself. And then I think it will be better if you mark and then you correct by yourself, right? And then you send another you send the copy to me again, because I want to analyze what of the question that you still do wrong. Okay, so we can have a look on this one. Okay, because I just read that you are not really familiar with the chapter 21. Okay, which is the biotechnologies. So this is the example of question which is from the past year. Okay, and then I do send you in this group. Where is it? Here. See this one, chapter 21 biotechnology answer. You can read all the answer first. Okay, so this is the chapter 21. Okay, let me show this one. Okay, which is about biotechnologies. Okay, the biotechnologies. Yeah, so that's why biotechnology, right? They always use the bacteria. Yeah. So the reason they use the bacteria is because that bacteria, okay, they will do what? Most of bacteria, they will do the asexual reproductions, which is the benefictions. So that's why asexual reproduction, right? They will just require a shorter time, lesser energy being used, and then they can divide in a very short time, in a very short periods, right? So that's why they are actually very fast. They can divide very fast. This is their greatest advantage because they can divide fast. So if they can divide fast, they can produce a large amount of product in a specific periods. Okay, so this is their feature. Okay, so explain why the doctor gives antibiotics. Because the antibiotic, right, is the one who can kill the bacteria. So you should mention that how antibiotic kill the bacteria. Okay, at this round, maybe you can tell me how the antibiotic can kill the bacteria, by what kind of mechanism or method. Antibiotic, they can target on which side on the bacteria, which part of the bacteria, which part on the bacteria. So remember how your bacteria look like. So now you tell me the antibiotic they target on, yes?
A wall.
Cell wall, correct, which is a peptidoglycan cell wall. So how they will do, you can say that antibiotic they just target on the cell wall, right? You can refer to here. I think I put it here already. Okay, this one, see? These antibiotics, right? They will just target on the cell wall. Okay, so this bacterial cell, they have a thick cell wall, and then they will rupture. Okay, how they rupture it? Because the penicillin, right? They can penetrate the bacterial cell wall by making a hole. So if there's holes, right, they will cause the contents leak out. Okay. So this one. So how can write the keywords? For example, right, the penicillin. Okay, so know that penicillin is a fungus, right? Penicillin is a fungus. So this kind of fungus, they will produce their product known as penicillin. So this one, fungus, they can produce the penicillins, which is the antibiotic. They can kill the bacteria. Okay, for example, they can kill bacteria. So how they can kill bacteria? They just interfere with the building of the cell wall of the bacteria, cause them to rupture and die. So the bacteria, right, they will undergo lysis. Okay, they will just lysis because the internal content leak out already. So that's why they will have lysis. And then what is very important is that you should complete a full treatment of antibody, right? So it's because that you want to prevent the development of the antibody resistance bacteria due to the natural selections. Okay. I think I do send you chapter 15 or not. Sometimes they would like to ask together in chapter 15 now, okay, which is this one. So this is not for chapter 15, okay. So and that's why I just remember that whenever you don't have time, right, it's okay if you don't read the long road, but you should read all this this quiz card, okay, with question answer. Yeah, so this is the thing that you can write, okay, it's just similar. And then yeah, so just remember that only antibody they can kill the bacteria, not virus, right? Antibody cannot kill virus. Why they cannot kill virus? It's because the virus itself, they do not have cell wall. There's no cell wall, so that's why they cannot target by antibody. Because antibody, they will be only target on the cell wall. Yeah, and then this one, they're actually referring to here, like how the antibody resistance is being produced. Okay. So this one, okay, explain how the population of bacteria become resistant to an antibiotic through the natural sections. So this is due to what? This is due to the bacteria, they can undergo natural selection. So that's why if you if you didn't complete your antibiotic treatments, right, what will happen to you? So there will be some of the bacteria left in your body, right? And then this bacteria will not able to kill by the antibiotic. Because this bacteria already developed resistance to antibiotic. So they will not being killed. So why this bacteria able to develop resistance? Because they carry the resistant gene. They carry resistant gene. So this resistant gene, right, either they just come from the mutations, which is mutation happen inside the bacterial genome. Okay, because mutation is a kind of random genetic mutations in the bacterial cell, so that's why they will acquire resistance, okay, in their gene. So this is a resistant bacteria. Okay, resistant bacteria, they just carry the genes which is resistant to the antibiotic. Yeah. So when the antibiotic is being used, right, they actually kill the non-resistant bacteria. Okay, and then usually all of the good bacteria, right, they are usually the non-resistant bacteria, because usually the good bacteria, okay, when you talk about bacteria, especially in your intestines, so Adam, I think you know that your intestines, right, they consist of which variety of bacteria living inside your gut. Okay, so that's why they will just create the selection pressure if they just using a new antibiotic drug. So usually the non-resistant one, they will be killed. They are being killed very easily. So the resistant bacteria, they just survive because they are not affected by the drug. Okay, and then with reduced in competition for the nutrients, right, the survival multiply rapidly. So it's like whenever these resistant bacteria they can survive, they will undergo more asexual production. So that's why they can produce a large amount of progeny. And those progenies, they consist of the resistant gene also. So now you're able to produce a population of bacteria which is not sensitive to the antibiotic being used. Okay, which is not sensitive to the same antibiotic being used in the first time. So that's why what can you do? Okay, what can you do in order to prevent the antibiotic resistance happen? Can you give me an example? What would do? What happened now? Okay, what happened if the medicine we use already developed resistance to the yeah? So what happened if the bacteria, okay, is already developed resistance toward the antibiotic? So what you can do? What's the method? You can change another antibiotic type, because you have a lot of antibiotic can be used, okay, which is in the different group, different category, but they are all belong to the antibiotic. Just their active ingredient also different, okay. Each antibiotic, right, they have different active ingredients, different kind of medicines. Yeah. So where is it? Okay, so it's like if the question asks about this one, suggests two ways the individual they can help to prevent development of the antibody resistance. So they will only take the antibody when they are absolutely essential. So just take antibody when you are needed. But usually, right, when we go to visit doctor, doctor will also prescribe us with the antibody. So when the doctor prescribe you with the antibody, right, you should try your best to complete all of the antibody dose. Okay, so always complete the entire prescribed course of the antibody. Even though you feel better after a few days. So that's why, even though right, you are recovered from disease, but you still need to eat continuously after finish. So this is to ensure that all bacteria inside your body, they are being killed. Okay, so that's why you need to like increase the dose also. Okay, so that's why what is the usage of the antibody in the agriculture? So antibiotics are often used in farming. Okay, farming to prevent the disease in the close quarter. This will create a session pressure that will increase the incidence of the antibiotic resistance, which can spread to humans. Yeah, so that's why what's it like farming? Okay, farming this one, okay, which is livestock farming, yeah, which is for my industry, okay. Our industry is like, I mean my industry, okay, is like rearing pigs and chicken, which is most of the Chinese people, right, they will eat the pork, they will eat the pork and also the chicken meat. So that's why we call it livestock. So usually, right, antibiotic also being used in the livestock. Okay, for example, antibiotic is being used in treatments for the sick animal, like in the sick pig or the sick chicken. You know, chicken or pig, they can be sick also. They can be sick, feeling not well, didn't eat or feed, just like in the humans, because they are animals. So it's like when the antibiotics are being used in farming, right? So they are aimed to prevent disease in the close quarter. So this will create the session pressure that will increase the incidence of the antibiotic resistance, which can be spread to humans through the food chain and the environments. So it's actually very dangerous for us also. If you eat the products, okay, especially the meat products, which is the meat products might contain the residue for the antibiotic. Yeah, they might be contain the residue for the antibiotic. So when you eat the meat, right, you eat the antibiotic residue also. So the antibiotic, okay, slowly, if you consume more and more, more antibiotic residue enter to your body, and then you are not realize it. So that's why when the antibiotic enter to your body, right, they can kill the bacteria inside your body. But if you didn't like, if you cannot be too frequent, because if you prescribe the antibiotic too frequent to the animal, right, and then especially for the treatment purpose, so what will happen is you can create the meat product which is very high residue for the antibiotic. So when the humans, okay, or the consumer, they eating on that specific meat product which is very high in the session pressure, right? Okay, so this will creating a session pressure. Yeah, this will create session pressure. So they will increase the incidence of the antibiotic resistance, which can be spread to human. Yeah, that's it. So that's for the chapter 21. So that's why you want to prevent the development of the antibiotic resistant bacteria. So that's why you need to complete full treatments of the antibiotic. And then for this one, okay, yeah, so you know that the penicillin, right, is come from the fungus. And then this is the thing that you should take note of it. Okay, that's what is this? Biofermenter. Okay, this is a biofermenter, and then so you should see, if you have a look, right, which is a label, okay, which is a label, which is from the air inflow, they will pass through the fans in the middle, and then like this. Okay, this one. Describe how the fermenter can be fertilized. So use the high temperature, high pressure to sterilize the UV radiation and gamma rays. Because high temperature, right, if too high temperature, they're able to kill the bacteria. But the resistant bacteria still not being killed, but at least they kill some of the part of the bacteria. And then for this one, you need to compare about the structures, okay? So water jacket, right, this P. is to maintain the temperature. This one, water. Okay, to maintain the temperature. Yeah, so that's why how you can sterilize the fermenter is you are using the high temperature, high pressure, UV radiation, gamma rays. And then this one, okay, so this is, you can try to fix by yourself. So S is the paddles, right? Okay, so S is the, we call it this one, paddles. So what is the function of paddles? Okay, sorry, let me open aircon. I feel so hot now. Just let me for a while. Okay, so now we continue. So what's my paddles? Paddle is just like is a kind of the equipments, okay, found in the fermenter. They will just turn around in a circle. So this is a paddle, okay. They'll just like turn in a circle like this, non-stop. They'll just rotate. They will just rotate non-stop. So they will just keep the nutrients moving in uniform mixtures. So they want to ensure that all of the nutrients in the in the medium, right, they are being evenly distributed. Okay, so P is what? Yeah, P is a water jacket, right? This one, water jacket. So this water jacket, okay, they can help to maintain the temperature. And then Q is a neutral inlet. So where is it? So this is a Q. Okay, so this is a neutral inlet. Can see? So this neutral inlet actually is the hole. Okay, so that's why the water that can come inside. And then the props. Okay, the props. So prop is just a sensor. Yeah. So this is a prop. Okay. So this is sensor. And then air supply oxygen for the aerobic respirations. Yeah, that's it. Okay, so you need to know about the structures. Okay, you should know about the structures and the label. Okay, the label. Yeah, and then for example, this U and T, right? Okay, air supply. So this one, number U, this one U, right? They are actually talking about what? They say that supply for the cellular aerobic respirations, supply the oxygen. So it's like they need to allow the air come inside. So when the fresh air comes inside, right, they can bring more oxygen inside. And then T is outlets. T. Yeah, so T is the outlets. And then this one, what we do is, where's my thing? Yeah, T is the outlet, right? Okay, so they will allow the collection of all liquid containing penicillin after fermentations. Yeah, so here's the answer. Okay, so what happens to the liquid containing penicillin after it's collected from the fermenter? Okay, so penicillin is kind of the antibody, right? It's being separated from the fermenter. So there's all impurities in the penicillin, right? They are removed by the purifications. And then, okay, for this one, okay, number 3. Yeah, number 3. So I think you should able to know how to write the nutrients, the role of the nutrients in the body, right? Now you already know. And then this one, describe in the detail how the enzyme functions to digest the proteins in the alimentary canals. Yeah. So like you need to starting from where? Stomach. And then in the small intestines. So Enzyme, they are known as what? They are known as biological proteins, okay? So the substrate will bind to antisense enzyme with the complementary shapes. And then enzyme structure complex is being produced, okay? It's being produced. And then enzyme remain unchanged at the end of the reactions, and the proteins are break down to polypeptide change. So actually this keyword are being listed in the answer, okay, in the mark scheme. But you can just have a read. This is just like chapter 4 also. And then this one, okay, this show small or large intestines. Where's the arrow pointing at? The arrow pointing at small intestine or large intestines. Want to have a guess?
Large intestine.
Yeah, this is the large intestines, which is shorter, okay? I hope you still remember the function of large intestines. They can help to absorb water, mineral ions. So explain why the lactose is not being absorbed by the small intestines. And then they tell about what? Oh, okay. So you know that why the lactose is not being absorbed? It's because lactose is what? Lactose is disaccharide, which is a bit large in particle size. So that's why they cannot pass through the intestine villi. Because intestine villi, I mean the intestine villi, they only require monomer. Yeah, only monomer they will absorb, which is a glucose. So that's why you are required enzyme. Okay, you are required enzyme. And then this suggests a danger of the health of several diarrhea if not treated for a long time. So what we call the health effect is it will cause loss of water, losing of water, reducing the blood pressure, reduce water pressure in the blood, and failure of kidney metabolism. And then for this one also, okay. So this show about graph, okay, this show about graph, which is a hydrogen gas. So this hydrogen gas, right, is being produced by bacteria that digest lactose in the large acids, which is around the same thing, okay, same thing. Okay, I think I start to discuss from 2023. Okay, I think we just go starting from behind first, okay, we discuss starting from 2023 first, okay, because 2016 is a bit old already. Yeah, I think we better start from here, okay, 2020. Here, okay, what is meant by biofuel power plant? What is meant by biofuel power plant? So, what is the source of the biofuel in the plant? For example, the corn, okay, the corn. And then this one, biofuel in the power plant. So that's why if you want to generate the biofuel, right, you have what? Crop waste, forestry waste, and the giant red plant. And then you have those waste, and then those waste actually they consist of the material. They consist of a material which is they are also important for growth. So that's why if you have these three kind of the waste, and then I will ask for the this one pre-treatments for the biomass, and then the complex carbohydrates released from the biomass. Okay, so that's why you know what's about biomass? Biomass, right, referring to the living organism. So after they die, they will become biomass. Okay. So we have crop waste. So for example, right, the crop waste consists of what? The crop waste are okay, they consist of the I mean the corns, right? The corns. Or the soybean meal. Yeah. And then the forestry waste, right? Okay, this one. Forestry waste, they consist of, for example, the fossil fuel, yeah, which is from the dead animals, dead plant. So we require pre-treatment of biomass. After that, you will have the enzyme, okay? Yeah. So after the carbohydrate releasing from the biomass, right, break down by the enzyme. Yeah. So and then what the enzyme will do? Enzyme, they will help to break down the carbohydrate into small pieces. And then after they just break down into small pieces, right? Now the monomer itself, the monomer inside, they can mix with other sugar or including the glucose. Okay, sugar or the glucose. And then fermentation by the yeast, right? Yeah, which is become ethanol and biofuel. Because right, just imagine this way. So what is biomass, right? Biomass is just like this. Okay, so I think you can have a read first. Biomass. I put that here already. Biomass. But it's very rare to come out in the biomass. This one. So see, this is we know as the biomass or the biofuels. It's almost the same thing. But we talk about the biofuel, right? Okay. So the process is just like first starting is the yeast, okay? So yeast are using the sugar as a food source. So that's why whenever there's an anaerobic respiration occur in the yeast, right, they'll produce ethanol and the carbon dioxide. Okay, so to produce a biofuel, you actually need to break down the plant materials and mix with the yeast. Okay, so yeast are are using the plant materials as a substrate in the anaerobic respirations. Okay, and then you know where you can get the substrate from. Where you get the substrate from? Where you get the substrate from? You get the substrate from the ingredient that you use. For example, the crop waste, forestry waste. Yeah. I teach you here already. Sorry. I show the different one. So the yeast, okay, just remember that whenever you want to do the anaerobic respirations, right, yeast is the most commonly being used because yeast they can do the both. Okay, actually yeast they can do the sexual or the this one. Yeah, but generally right they will do the sex this one the asexual productions, which we know as well. They undergo cell divisions. Okay, they will undergo cell divisions. So whenever the yeast use the sugar as a food, right? So where is you get the sugar from? You get the sugar from the breakdown of the carbohydrates. Okay, you get the sugar from the breakdown of carbohydrate, and then this carbohydrate they are from the plant base. For example, corn. You know, corn is one of it. So it's like it might be due to the crop waste. Okay, crop waste or forestry waste. Okay. At this one you have a read on this first. Okay, and then wait for a minute. For 1 minute, I need to settle thing. 1 minute. Okay, I'm back. Sorry, just like I'm answering a phone call. Okay, so it's like this. So how can you make the biofuel? Is from the plant, okay? Plant. What kind of plant? For example, the corn, okay? Corn or the wheat, any kind of plant. So the plant, okay, they will undergo the fermentation because the plant inside, for example, the corn, right? What are found inside the corn? At this one, inside the corn, right, you have what kind of nutrient molecule? Proteins, fats, or carbohydrates. Inside the corn. Now you can see inside this corn, the yellow corn, consider what kind of nutrient molecule? Proteins, carbohydrates, or fats. At the end, you will get the ethanol as a byproduct. So think what kind of nutrient molecule inside the plant? What's the answer? Yes? You're thinking, right? It's okay, just give me your answer. You think is carbohydrates, proteins, or fats are found inside the corn. Yes, the answer is carbohydrates. Okay, the answer is carbohydrate because when you look at this, see, carbohydrates, okay, which is your biomass, the biomass, the plant, or the waste, okay, or the waste. So they will contain the carbohydrate inside. Okay, what kind of carbohydrates? Cellulose, starch. Do you remember? So those plants, right, consist the carbohydrates, and then you are required to use the enzyme to break down the carbohydrates. So that's why, for example, right, if they want to process the corn, if you want to process the corn, see the corns, later they'll become the ethanol, okay? Later they'll produce the ethanol for the biofuel, right? So what they can do is they'll just grind them in small pieces, and then they add it with the enzyme. Yeah, they will add it with the enzyme. So the enzyme, they will just break down the complex carbohydrates inside the biomass, right? From what? From the glucose, okay, become the glucose monomer. Yeah, so that's why they will release sugars, including the glucose. So glucose is just one of the product being produced, right? Yeah. So that's why the glucose, okay, glucose is a source of the yeast fermentations, which is whenever the yeast they want to do the fermentation, right? They are required input of the glucose to do this. Yeah, you require the glucose because glucose are involved in what? Glucose, they are involved in the anaerobic respirations, also in the aerobic respirations. So how the glucose can produce the ethanol, which is by the aerobic or anaerobic. How the glucose can convert become the ethanol, release the ethanol. Is the aerobic or anaerobic? The answer is the anaerobic equation. Okay, so it's an anaerobic process, and then here's the equation. Yeah, so you should have a look, okay, the glucose, because they ask this kind of question before they will ask you to state down, okay, they will ask you to state down like the equation. They will ask in this way: state the balance equation for the anaerobic respirations. So that's why this is the answer, okay. C6H12O6. Put an arrow. C2H5OH. Remember to put 2 in front. 2 in front CO2. You need to balance the equation. Yeah. So that's why this fermentation, because yeast fermentation is a kind of the anaerobic respiration. So that's why yeast fermentations, yeah, they're known as fermentation because most of fermentation, right, they are being conducted in the condition without oxygen supply. So this process, okay, will help to make the biofuel. For example, like you can have a look at this. So that's why they will just grind the plant material like corn into the small piece, and then they'll just add it with the enzyme. Yeah, and then the enzyme just break down from the carbohydrates, become the glucose, from the glucose, and then they will just add this the glucose, right? They will allow the anaerobic respiration to occur. Okay, so that's why they will produce the ethanol as a product. And then later they will need to extract out the ethanol. Okay. And then they will need to purify the ethanol also because they want to ensure that only ethanol is being isolated out, not other kind of component. So each cell, okay, they'll just use a plant material as a substrate in the anaerobic respiration. What does this mean? Because plant material consists of the source of carbohydrates, which is the cellulose starch, right? So that's why they will be break down by the yeast to produce the ethanol as a byproduct. So once the liquid is being separated, right, the water is removed to leave the concentrated solution of ethanol. Okay, so you clear about it, right? How the biofuel is being processed. You need to be very clear, which is from the plant material, break down by the enzyme, mixed with the yeast. And then the yeast, right, you can use the glucose as the source of the anaerobic respiration and produce ethanol as a byproduct. So that's why they will undergo yeast fermentations after the glucose is being produced. See, write the equation. So if you know how to write the equation already, so which is just copy on the equation that I showed you just now. Okay, and then this one suggests the environmental advantage of using ethanol as a fuel. So what's the advantage if you use ethanol? Okay, even though actually, right, the ethanol, okay, ethanol is a biofuel, right? Especially ethanol is being used to make the petrol. Okay, petrol, like usually the car, they will inject, they will help to pump in the petrol into the car. So that's why the car able to operate. Okay. So even though nowadays, right, they already they already created with the electrical car, okay, electric car, but they still feel that the car, which is driven by the this one, okay, driven by the petrol oil. Yeah, because petrol oil also example of the biomass. Petrol oil also might be extract from the plant also, right? So that's why what's the environment advantage, right? If you recycle the ethanol as a fuel, so you can use the waste. Okay, you can help to clear the waste, which is major of the waste that they're producing from farming and forestry, and then you can help to reduce the fossil fuels, cause less pollution to the environment, because you don't need to like burn them to dispose them. Because most of the garbage, right, how they're being disposed is they are being burnt. So you have the open combustions to release more carbon dioxide. And then this one, okay. Giant reed plant, they are monoculture. And then this advantage, okay, this advantage of growing giant reed plant to provide biomass is if you're just growing one plant, right, in the area. The area is just have a very large space, very spacious, very wide space, and then you just use the area to plant one plant species, okay, which is this one, giant reed plant. So that's why it will cause what? It will reduce in the genetic diversity. Okay, and then it will cause some of the animals, right, that depend on that plant to survive, but there's no plant, and there's no that specific plant because all already being occupied by the growing giant reed plant. So that's why you have a loss of genetic diversity, a loss of species diversity, because it will cause some plant species, right, reduce in their number. So if the plant species reduce in number, and then there might be a source of food to the animals, right? So that's why the animal will not get enough food. So that's why they will just die, and then you are actually reduce your species diversity. And then the plant will just grow in the monoculture to the disease. Yeah. Okay, so that's why this is a disadvantage. Major, the most important thing is you are actually reducing the diversity. And then this one, yeah, they show about the graph. Okay, so xylose and the ethanol acid. So these cannot use the xylose, and then the ethanol acid is toxic to the yeast. And then you have this three graph. Okay, concentration of the xylose and the concentration of the ethanol acids. And then the concentration of the ethanol at here. See, here, here, here. Okay, so because the line already, and then what they say about this one. Okay, they also describe the result. Describe the results in the figure 4.2. So that's why you can see like the general trends, right? So you can have a look that which is only here. It is like increasing. Oh, sorry. Which is only here, right? Here is increasing in the concentration of xylose or ethanol. Yeah, so that's why you should say that this one, right? Xylose and ethanol acid. Yeah. So you can see this xylose and ethanol acid, right? They are actually decreasing. Yeah, they are decreasing. Okay. And then who will increase? The concentration of ethanol, right? They will increase. Yeah. So because they ask you to describe the result only. Describe the result, no explain. Okay. Understand? Describe the result only. How many marks? 3 marks. Okay, so whenever the question asks about describe means like you need to describe the trends. Okay. So describe the trend of graph. Either the graph is like increase, right, or decrease, or even a plateau. Okay, we don't plateau. Around that. Yeah. Or reduce. Sticky. So you just need to include your result, okay, result. And then you can compare between the xylose ethanol acids So that's why there's a steep decrease of the xylose and ethanol acids until 62 hours, and then there's steeper change between 6 to 12 hours. So it will increase the ethanol concentration to 44. And then there's small change in the concentration after 62 hours, right? So the xylose, they will just reach at the 90 hours. Yeah, so this one, they just show about this one. Yeah. Here. Okay, they are actually asking about here. So this is not very hard, just like you need to know how to describe it. Yeah, so later I'll just send this to you, and then please have a look for this chapter. Okay. Possibly, I mean, it's very high possible that might be this chapter will come up in the exam, which is chapter 21. Okay, so that's why and then for this one, predict the result that you would expect from the concentration of ethanol, right? So there will be slower rate of increasing of ethanol, okay, because it takes longer time to reach same concentration of the ethanol. Okay, so this is like this, and then I want to show you another thing, which is this one. I see this one, okay. 26, this 26. So that's why just remember that penicillin is the antibiotic. And then the one who produces penicillin is of course penicillin. Yeah, and then state the name of the group of medicinal drug, right? They include penicillin, which is an antibiotic. And then this is of course what? Production of penicillin, which is in the real life, right? In the real life company, which is making some new product, right? They actually do it this way. Okay, just similar. So that's why it's actually like step by step. Okay, so that's why this one. Yeah, the main feature of fungal cell, they are used to distinguish them from the cell of prokaryotes, right? So that's why you have the nucleus mitochondria, because and that's why fungal cell is a prokaryote or is eukaryote. Fungal cell is prokaryote or eukaryote. Fungus.
Prokaryote.
Fungus, prokaryote, are you sure? Fungus.
Eukaryote.
One of the kingdom. Yeah, fungus is eukaryote. So who is the only one belong to the prokaryote? Bacteria. Okay, you put friend, bacteria. Bacteria is only the prokaryote that you learn. Refer to chapter 2. I think I sent chapter 2 notes. It's okay. This one, chapter 2. See? This one. And then you should know it very clear, which is the difference in the structures for the prokaryote and the eukaryote. This is very important to know. So just remember that whenever they come out the structure difference, right, you should answer in this way. You can talk about nucleus. Okay, talk about nucleus, talk about the eukaryote, they have organelles, but not in the bacteria. So just remember, bacteria or the prokaryote is the only, bacteria is the only prokaryotes. Okay, it's only prokaryote. So see, you have animals, plant, fungi, bacteria, protists. So only bacteria is a prokaryotes. Other like animal, plant, fungi, and protists, right? They are actually eukaryotes. Okay, they are actually eukaryotes, which is like this. Okay. You just have a look on this, right? So I hope you help you to memorize easier. So actually you can see, right? In the prokaryote, there is no nucleus, no membrane organelle like mitochondria. ER also don't have, but they have a circular DNA. Okay, they have a pathway. They have a cell wall. Yeah, so that's why this one is step 1 by 1, and then say the genus name in the organism A, right? Yeah, so that's why this is the penicillin. Okay, and then for this one, penicillin is being produced in the fermenter, okay, and then a variety of nutrients X, Y, and Z, right, are mixed together and added to the fermenter in the step 1. So that's why what kind of nutrient you can add to produce penicillin? So just very simple. Most important is carbohydrates. So where you can get the carbohydrate from? You cannot write carbohydrate, okay, you can only write the nutrients, the simplest nutrient, glucose, or then or the amino acids, okay, or the amino acids. So explain why the nutrients are sterilized before they are added to the fermenter. So they want to stop the growth of microorganism. And then this one, okay, why the fermenter has a gas outflow pipe? Yeah, where's the gas outflow pipe? Oh, yeah, this one. We can find yeah, gas outflow pipe. I think it's here. Here, okay, here. Which is on the top. So they need to allow the gas being flow to outside. It's because they want to prevent too much of air stored inside. Yeah, because the function of gas outflow pipe is they will help to release the waste and carbon dioxide, any kind of gas. Yeah, so they help to reduce the pressure. Okay, and then this one, outline the events occurring from step 4 to 8 during the production of penicillins. Do I have a picture? Hey, I don't have any picture. Oh, sorry, I think I put it somewhere. I mean somewhere else already. But you need to identify, okay, step 4 and 8, right? So, step 4 is the reproduction of the penicillin before adding to the fermenter, and then you can inoculate into the fermenters, and then you realize that there's an exponential growth of the organism in the fermenter, like this, and then you need to purify the products. Okay, that's all. And then for this one, okay, let me show what we have restriction enzyme. So you see, this is the DNA. Okay, you have upper strand, lower strand. I think this is from the human DNA, I think. And then you use the enzyme to separate them to 2 strands. Okay, so what kind of the name of enzyme that you how to cut this one? What's the name of this enzyme? Enzyme 1. And then by using this enzyme, right, you can cut a small portion, and then you can see that there's a okay, so this is the one who need to join with another, yeah, by the complement-based pairing. So that's why, and that's why, what's the name of enzyme? Tell me what's the name of enzyme which they will help to separate the DNA molecule. Restriction enzyme. Is that okay? Restriction enzyme. Which is Caesar? Okay, so that's why describe of the effect of the enzyme 1 on the DNA molecule in step 3. So it helps to cut the DNA molecule to form sticky end. And then the sticky end, right, are complementary to another DNA molecule. So when you cut them, you can produce a sticky end, which is here. Do you know here is a sticky end? Yeah. So you can only use the restriction enzyme how to produce a sticky end. So this is the restriction enzyme functions. Okay, they help to cut the DNA molecule to form a sticky end. So this sticky end, right, they're actually very complementary to another DNA molecule. Okay, and then explain how the enzymes specific to the asset sequence of DNA base, right? So DNA base sequence, okay, they are complementary to the anti-cell enzyme. Okay, so this is the same thing. So just need to know is there will be 2 enzymes, restriction enzyme and the DNA ligase. So one is split, then one is separated. And then here is something from here. Later, I will just give you some homework to do also. Okay, I will give you chapter 18. You can, chapter 17, because we haven't finished about this also. I think last time we just finished up, but we are not discussing too clear. And then I remember I submit. I mean, I just remember I sent the homework to you, right, about chapter 17, but didn't get your reply. It's okay. I'll just resend again in the group, and then you can try to do it in your free time. And then later I can have a look on the answer. Okay, because you still have about, I think it's 9 weeks to go. 9 weeks to reach your actual exam. Okay. Okay, the function of chlorophyll is very common question also. This is from the chapter 6, the nutrition. They're talking about the functions of the chlorophyll. So chlorophyll is a photosensitive pigment. So what's the function is they will help to convert light energy to the chemical energy to make the carbohydrate or glucose. Okay, and then this one, state the name of the one mineral for the chlorophyll, right? Is a magnesium. Okay, and then this is just to compare the change in the pigments. And then for this one, okay. So both, right? No matter it's the red color or yellow color food. So the chlorophyll concentration, right, actually decrease in both. Okay, so unripe this one, unripe fruits, they contain the highest chlorophyll concentrations than the ripe fruits. So you can have a look, right? See, unripe more than the ripe fruits. Yeah. So that's why the lycopene concentrations increase in both. And then lycopene will increase from 0 to the unripe fruits in this data. Okay, so and then you can conclude that the lycopene change, right, is much larger than the chlorophyll change. So you see, right, they are not asking you to explain like why the unripe yellow fruits, right, they have a higher concentration than the ripe yellow fruits are. Okay, so that's why I just need to compare. Okay, they will just usually give you 2 types of data, and then you need to draw the table, compare it. Okay, and then one more questions which are here, I think. Okay, this we discussed with it before already, and then you only very clear know about what is the difference in the physical and mechanical digestions, right? And then here. Yeah, and then this one, because right, you are using, your biological powder, there's an enzyme inside your biological powder. So that's why they are not really resist the high temperature. You just worry the enzyme structure being denatured at the high temperature. So that's why the enzyme this side, right, they will just contain a specific shape which is complementary. Yeah. So 30 degrees Celsius is the optimum temperature. And then when reach about 50 degrees Celsius above, so temperature will increase, okay? The kinetic energy between enzyme and substrate will increase. And then later, the enzyme molecule, right, they just vibrate vigorously. Yeah, because they are increasing your kinetic energy. So definitely they will just move quickly. Okay, and then for this one, right, here. I think I show you the actual one, it will be better. Okay, I just show the actual one. It will quite blue. It's not too clear, the wording. If I put that here, I better show you the actual one. See, this is the actual one. Outline the role of bowels in the body. Yeah, so this is the function, okay, emulsification or neutralize. And then this one, yeah, this is 21 questions. Yeah, 2021 questions. So you can see, right, this is a bacterial cell. And then this bacterial cell, right, is being divided by the mitosis. Yeah. So it's an asexual reproduction. So DNA is one whole form of the double helix. And then DNA is copied to number of the loops. And the cell division is the same in each daughter cell. So daughter cells are genetically identical to the original cell. Okay, here. Yeah, I think that's all for this one. And then another question. Okay, see, they always come out the similar question. Again, what kind of enzyme to cut the gene on the DNA? So it's like actually restriction enzyme. So at this one, maybe you can refer to here. Okay, this chapter 21, you can refer to here. I think it's here. Okay, what is this? Yeah, actually I already put that here. You can have read again. This one, okay. So just remember that few process. So that's why you have a restriction enzyme, and then the enzyme will just cut on the human gene. They will also cut on the bacterial gene. So later the DNA ligase, right? See, DNA ligase is one enzyme that will just help us to fuse together, and then they will become the recombinant plasmid again, because the plasmid already consists the foreign DNA, which is not produced. Which is not found inside their body. They will have a foreign DNA. Okay, so that's why DNA ligase, right? They just join the gene and plasmid to form the recombinant plasmid. So means that after that you generate recombinant plasmid, right? You need to input back to the bacterial cell. Okay. Yeah, see, so just need to undergo about few points. So starting, you can the restriction enzyme. So the restriction enzyme, they are used to isolate the required genes, right? And then you produce sticky end. So I want to see this one. Restriction enzyme, sticky end, cut. Okay, cut on the DNA. And then so that's why you should write about like one is the insulin gene. Okay, one is the DNA of the insulin gene. Another one is the actual bacterial plasmid. So the plasmid and the isolated genes are joined together by the DNA ligase, right, to form the recombinant plasmid. So after they become the recombinant plasmid, right, they will insert into the bacteria. Okay, so they will just insert into the bacteria, and then the recombinant plasmid, right, is insert into the bacterial cell. And then when the bacterial cell reproduce, right, they will copy thousands of the plasmid. Okay, so that's why they produce a lot of new bacteria, which is they will consist the genome for the recombinant plasmid. Okay, this is the enzyme. And then for this one, some students conclude that the cell must be at least 6 micrometer in length before cell division that can occur. So describe the evidence against the student conclusion. Against, okay, means what? You write in the opposite way. Yeah, so because their conclusion is what? The conclusion is the cell must be at least 6 micrometer before cell division, but actually it's not, because even though the length, right, the length of the cell is not 6 micrometer, but it's only 4.4. But they're still able to, grow, increase in their size. All right, due to the cells, they still are able to undergo the cell divisions. Okay, that's all. 22, right? And then here. This is the part that I think in the variation topic. Okay, so sorry, why bacteria is being used, right? The main reason is if you use a bacteria, right, you would have no article issue. And then they'll just produce rapidly small size. Okay, and then you know that insulin, right? Insulin is kind of hormones. So that's why it will be produced by the pancreas. So what happens if the pancreas being destroyed? You have type 1 diabetes. So at this one, you have about 2 types of diabetes, right? Type 1, type 2. So that's why type 1 will be more severe, because type 1, they will have the destruction of the cell. Yeah, the pancreas cell. So that's why if you have a destruction of the pancreas cell is being disrupted. They are unable to, they are unable to because the pancreas cell is being destroyed, right? So there's no insulin productions. So if there's no insulin production, right, what will happen to the cell is they are unable to carry out their function. Okay, so function insulin is they will help to maintain, reduce the blood glucose concentration. How they reduce is from the glucose glycogen. Okay, and then this is the phase, PQRS. So starting from here is a lag phase, exponential phase, stationary phase, and then number 5 is a death. Okay, death. And then why the bacteria did not grow in the fermenter for longer than 5 days? Well, it's too long already. You cannot allow your bacteria to grow more than 5 days. Okay, so it's because that if you grow more than 5 days, right, and then there will be some factors not being observed, which is there any space enough? Is there any populations or competition happen? So, but what we know is, because there will be fewer and fewer nutrients inside that container. Not enough oxygen is being available. So that's why they will build up a lot of toxic substance. Yeah. So because the building of the toxic substance in their surrounding environment, so some of the bacteria they will not grow well in the fermenter more than 5 days. Okay, so that's why you know what's the reason is because they don't have enough nutrients. Okay, and then you to build up of the toxic substance. Okay, last one. Okay, I think I discussed one more question to you again. Okay, and this one you can spend some time, right? You can read about this one. Okay, please read about this one. This one. Is there any important question? Okay, so for another question, right, which is this part, they will ask about the application. For example, what is the application of the yeast fermentation in the bread? So that's why they will able to produce a gas. Okay, see, the carbon dioxide gas is being produced in the yeast fermentation to make a dough. Okay, a big dough. Yeah, and then this is the what kind of products they can make use of microorganisms. So that's why you can use the biofuel or penicillins or bears, up to you. Okay, and then this one, why yeast is being used in the bread making process is because yeast, right, they will release a carbon dioxide. Yeah, and then this carbon dioxide is very important because you are requiring this carbon dioxide. They can help to increase your height of the bread dough. Okay, definitely, because before you produce a carbon dioxide, right, you need to have a fermentation ongoing first. So, which is a yeast fermentation, and then the yeast they will get what? Yeast they will get the sugar, okay, the glucose. So that's why the yeast they will produce the enzyme, and then this enzyme they will break down the carbohydrates, become their monomer. Okay, and then this one, okay, describe how penicillin is used to make the antibiotic penicillins. Yeah, because they just grow in the fermenter. The air is bubble through 4 aerobic respirations, provide sugar and acids. I think okay, and then for this one, see, describe the process of using bacteria in genetic engineering to produce human protein. So the process is you should first isolate it. Yeah. So you should isolate your gene, human gene. And then later you use to cut. You cut the gene with the enzyme, okay, the bacterial enzyme. And then you can produce a sticky end. And then another from the bacteria also cut the bacterial enzyme. And then the DNA ligase, right, used to join the plasmid and gene to form a recombinant DNA, okay? And then the plasmid are inserted back into the bacteria. And then bacteria they will reproduce and produce insulin already. So which is called, we are known as the recombinant bacteria, because the recombinant bacteria they consist of the recombinant plasmid, which contain the human insulin gene. Yeah, and then the part in the fermenter, okay, for example, the steam, okay, yeah, they can release the steam to kill the pathogens and reduce contaminations. And then the 3 conditions inside the fermenter, yeah, so you can measure, okay, you can use a probe to measure the change in pH, temperature, oxygens. And then this one, okay, why the glucose and nitrogens were added to the fermenter? Because as a source of the respiration. And then this, why is it important to stir the content fermenter? Because we want to ensure all of the solution is mixed evenly. See, for the even distribution of contents, prevent clumping of content. See, clear cut the answer. Yeah. So this one, okay. Okay, and also I hope you know how to look at this picture, right? Do you know what's meant by this picture? This picture right show that this is the agar. Agar is something like a small plate. This small plate only found in the laboratory, because when they want to culture the, for example, fungi or bacteria, right, they will just use this kind of dish to culture it. But this provides them with sufficient nutrients. Yeah. So this is the thing, okay? And then if you have this one, number 2, right? So and then this is the penicillin here. Okay? Yeah, here. Okay? Are you still there?
Sorry, left by accident.
Okay. Sorry. Just for 2 or 3 minutes only. Yeah. So that's why this is the area which contain the antibiotic penicillin at here. This is also the area. And then what will happen is, what is this white area? What is this white color area? And then what is this transparent black color area? This means what? This means the zone. See, the zone where the bacteria are growing. Yeah, they're just growing at here, but they are not growing at here. So here there's no bacteria growing inside here. But if you refer to this plate, right? This plate, they have all bacteria just growing until here. So now, and that's going to tell me, you have plate number 1 and 2. You tell me which plate they have the bacteria, and then this bacteria already develop resistance to the antibiotic. Which plate of bacteria, right? They already develop resistance toward antibiotic. Plate number 1 or number 2?
1.
1. Okay. Why you choose 1? Okay. My question is, which plate, right? They have the bacteria. This bacteria develop resistance to the antibiotic. Okay. If the bacteria, if the bacteria develop resistance to the antibiotic, right? The bacteria can be killed by antibiotic or not. If the bacteria, okay, if the bacteria develop resistant to the antibiotic.
Can be killed. Can be killed.
Cannot. Resistant means what? 抗药性. In Huai, it's 抗药性. Cannot be killed. 抗药性. This one. So just remember, if the bacteria develop resistance to the antibiotic, right, it will unable. Which is that? Because resistant bacteria cannot kill by the antibiotic. So that's why number plate 1 and number plate 2, right? So you can see the plate 2, actually the plate 2, the bacteria inside the plate 2 already develop resistant to the antibiotic. 抗药就是说已经对这个抗生素产生抗药性了。So that's why the antibiotic in the plate 2 unable to kill the bacteria. So you can see the bacteria, right? They'll just grow until here. But in the plate 1, right, you can see a clear zone, a transparent clear zones around them. And then the bacterial growth is until here. So this means what? This means that the antibiotic in the plate 1, right, they still able to kill the bacteria in that plate 1. So there's no bacterial growth inside, see? Because the bacteria in this region, right, they are being killed. They are being killed by the antibiotic. So it means that bacteria, they are still sensitive to the antibiotic. Understand? And that's well,
yeah.
Bacteria, they are like sensitive, okay, 敏感 to the antibiotic. 敏感 means what? Sensitive. Yeah, sensitive 敏感. So which is the bacteria, they can kill by antibiotic because they are still they are still not develop resistant yet. 就是说他们还没有产生抗药性 not develop resistant yet. So they can be, okay, they can be killed by the antibiotic. And then one thing, okay, whenever the question asks about how the antibiotic resistant bacteria are being developed. So what kind of process? Is from the artificial selections or natural selection? Which one? How the antibiotic resistant bacteria is being produced? From the artificial selections or natural selection? Which one? Hi. How you can able to produce the antibiotic resistant bacteria? Is from natural selections or due to the artificial selection? Which one? The answer is natural selections. Okay, you can refer to this chapter 15, chapter 15, okay, which is this one. Yeah, here. Okay. I want to show, okay. You can refer to this note. Okay, this note. This chapter 15. Where is it? Chapter 15. Okay, I show you how the how the bacteria develop resistant toward the antibiotic. It's because due to the natural sections. You can just imagine like this way. See, this is in the chapter 15 drug. Okay. Just need to memorize this one. Okay, I just put it here first, and then I'll just send to you again. Yeah, antibody resistant. Okay, for your information, chapter 15, right? Only you need to study about the antibody resistant. You no need to study about alcohol drug, not include in your syllabus. Morphine, heroin, not include syllabus. So that's why you see chapter 15 are just very short. You just talk about define the drug. Okay, what's the antibiotic? Why antibiotic they can kill bacteria? Why they cannot kill virus? Okay, how they kill the bacteria, and then how you can develop the antibody resistant. How can you prevent the antibody resistant? Okay, so just very simple chapter. Okay, so see, this is the picture. Okay, so just imagine in your body. Okay, actually you have a lot of bacteria just living in your body, especially in your intestines. You know, your intestines, they consist of great variety of the bacteria, and then you have a good bacteria or you have a bad bacteria. The good bacteria means that they will not cause harm to your your body. Okay, they will not cause harmful effect to your body. This is known as good bacteria, and then they can help in your food digestion process, which is sometimes this bacteria they can secrete out some kind of enzyme or acids. They can help the food digestion process, which is they can help your food to being digest. Improve. They can help to improve the digestion process. So that's why they can help your body to absorb more nutrients. So this is the role of the good bacteria, which is they are in the red color. Oh, sorry, they are in the yellow color. Sorry, it's not yellow, green color. See this green color. Okay, can you see this green color? This is called what? Healthy gut bacteria. Okay, healthy gut bacteria. So that's why we call population of bacteria in the gut. Gut means intestines. Okay, so some of bacteria, not all, just some, and just very few bacteria. Just a very few bacteria, they will consist of what? They will just consist of resistant gene. For example, this bacteria, okay, I put, okay, I think I put, what color? I should put, okay, I think I put this one, yellow color, okay, yeah, here. Okay, so just imagine this yellow color, right, refer to the gene. They have a resistant gene. Okay, resistant gene against the antibiotic. And then you have this one. Okay, so when you are exposed to the antibiotic, so antibiotic, right, they will kill all bacteria. Okay, sorry, they will not kill all bacteria. They will just kill the bacteria which develop, which are still sensitive, to the antibiotic. So that's why you can see the green color, which is a good bacteria, and then the red color. The red color actually is a bad bacteria, but this bacteria, they will not carry any resistant gene. So they still left the purple color bacteria. Okay, see, purple color bacteria, they are not being killed. So that's why These purple color bacteria, while not being killed, they will still undergo asexual productions. They will divide and produce more and more progeny. So those bacterial progeny, they will consist of the resident gene. So that's why over the time, right, this purple color bacteria, which consists of resident genes against that antibody, because the antibody unable to kill the bacteria which carry the resident gene. 抗药性基因, okay? Resident gene means 抗药性基因. So see, with the reduced competition for the nutrients, so antibody-resistant bacteria, they can multiply, forming a larger populations that are very difficult to control. Okay, so just like this. So that's why you need to finish your antibiotic course, okay, which is if your doctor, okay, prescribe you with about 10 tablets for the antibiotic, right? You should finish all of the tablet. You cannot only eat half, okay, and left half still haven't eaten. It's because it's very important that you should finish all the antibiotic medicines. You want to ensure that it's a enough dose, okay, sufficient dose to kill all bacteria. You want to ensure all bacteria is being killed, especially for the pathogenic bacteria. And then usually the pathogenic bacteria, right, they'll just carry the resident gene. And then usually good bacteria, they will not carry the resident gene. So that's why when you use antibiotic, right, you just kill all bacteria. You hope to kill all bacteria, including the good bacteria and the bad bacteria in your gut. In your gut. You just kill everything. Okay, so after the good or bad bacteria die, right, there will be no more bacteria in the area. So you can cause the bacteria to regrow again. So you just don't want the antibody-resistant bacteria to regrow. You only want the good bacteria to regrow again. Understand? You want to encourage the repopulation of the good bacteria. So, for example, like in this way, okay, if you just finish about 6 tablets, right? For example, if the doctor give you 10 tablets of the antibiotic, just meaning about 6 tablets, right? So that's why you would left some of the pathogenic antibiotic, pathogenic bacteria, which consists of the antibiotic resistant gene not being killed. So that's why they will have a chance to reproduce. But if you complete all tablets of the antibiotic, you eat about 10 tablets, okay, you just finish all tablets. So you can ensure the dose is sufficient to kill that bacteria which carry the antibiotic resistant gene. Because you have a high dose, okay? You have a high dose. High dose, they can help to kill the bacteria, the bad bacteria with the resistant gene. So by this way, right, you can prevent the issue of the antibody resistant bacteria. Okay? You know what I mean? Can you understand?
Yeah.
Yeah, so it's like, okay, how you able to limit the antibody resistant bacteria? This is a very common issue nowadays. Okay, especially, right, we have lesser choice of antibiotic. Because we don't have enough money to develop a new antibiotic. Okay, antibiotic, right? Okay, I know one of the antibiotic that we learn is what? Penicillin. And then it's the most common use. Actually, penicillin, this one. Penicillin, right, is the first type of antibiotic being created. And then there will be another antibiotic, for example, what kind? Amoxicillin. Okay, so see, you have about like antibiotic types, and then see, you have difference there. These are cephalosporin, macrolide, tetracycline, those kind of antibiotics. If you do medicine, right, if you study medicine or biology in future, you will learn more about this. Or you study pharmacy, I will learn this antibiotic type. So it's like you know that, okay, the doctor, right, sometimes doctor, they just give you a test, right? They will just test that your body, I mean the bacteria inside your body is sensitive or resistant to which kind of the antibiotic. So that's why if they detect that your bacteria inside your body, right, is sensitive to that certain type of the antibiotic, they just use the antibiotic. Okay, they use the antibiotic. For example, if they detect that your body is sensitive toward the cephalosporins but already developed resistant to the penicillin, right? So they will not use the penicillin again because they know that penicillin unable to kill the antibody-resistant bacteria in your body. So they will use another type of the antibiotic. Okay, for example, cephalosporin or macrolide, other type of antibiotic. So for the exam purpose, you just remember about this one. Okay, you should rotate the use of antibiotic. You cannot use the same type of antibiotic to treat the disease. because you want to avoid develop resistance. Okay, and then you should always finish a course of antibiotic treatments, and then you should don't overuse the antibiotic. Only use it when you need it. Okay, practice new hygiene. Practice good hygiene, and then develop new antibiotic. But this is very hard. Develop new antibiotic because you don't have. I mean, recently, okay, they don't have much money to develop new antibiotic. So that's why there will be lesser antibiotic can be used again. Okay, that's all. Yeah, so that's all for today. Yeah. Okay, and then this one, chapter 21. Okay, please spend some time, okay, to read on the answer. I think we learned it before already. So just that you can record it or not. Just read on the answer. Yeah, and then you can try, okay? Try to do the paper 6. Read on the answer for the paper 6 also. Okay. And then that's all. Do you want to try paper 4 again? Okay. Okay, I will just send you paper 4 to do again. Okay, I just send you 2024. Okay, that's all. Bye. Bye. Okay. Good night.